Sweet. G'day and thanks for joining me, Luke Florence, for this episode of the South American Mycorrhizal Research Network's Journal Club, What We're Reading. Today we're joined by Adam Frew to discuss his paper, which is titled, I'll just bring it up. It's titled Community Assembly of Root-Colonizing Arbuscular Mycorrhizal Fungi, Beyond Carbon and into Defense. So thanks for coming along today, Adam.
That's all right. Thanks for having me.
Could you start by introducing yourself and giving us an overview of the paper and highlighting how your research has led you to develop the theories that you propose in this paper?
Yeah. So I'm Adam, as you said. So I'm a lecturer in mycorrhizal ecology at Western Sydney University within the Hawkesbury Institute for the Environment. And what was the second question?
Just to provide an overview of the
The
paper,
paper,
and,
yeah.
and if you could also just discuss how your research has kind of led to the development of the theories that you propose in the paper.
Oh, yeah. Okay. So I suppose in doing that, I can give a bit of background of, I suppose, my research history as it is. A lot of when I first started working with arbuscular mycorrhizal fungi, so AM fungi, it was with a view to looking at their effects on plant defense. And as a lot of people or some people might know, there is quite a bit of literature on this, so on how and when the AM symbiosis does increase plant either resistance or tolerance to herbivory, specifically, is what I was looking at. And so for me, that was always kind of the aspect that's kind of where I started. And I imagine probably a lot of people who have started their research careers with mycorrhizas, they tend to start from the nutrient perspective, and maybe for many they're not quite so aware of the AM symbiosis and how that relates to plant defense. So I was always kind of looking at it from that angle, and then I suppose more as I've looked into different questions, I sort of started looking more at not just the effects of the mycorrhizal symbiosis on defense, but then what about the diversity of AM fungi or the community composition? Does that have an impact on defense, and does that vary? And then it was always kind of bottom up. So it's like the AM fungi or the AM symbiosis affects the plant, and how does that affect the plant's interactions with a herbivore or a pathogen? And so I suppose there was less looking the other way, right? So how a herbivore or a pathogen would then affect the AM fungal community. So that's kind of how I sort of came to ask these sorts of questions, I suppose, and start to think about, well, how much do we know about these interactions, so both ways, bottom up, top down, and how might they be functioning? How might they have come about? And so that kind of led to the sort of things we were discussing in this paper in ISME.
Cool. I guess I'll jump and have a look at your figure 1, and perhaps you could describe the carbon, summarize how the carbon constrained and defense directed hypotheses are, I guess.
Yeah. So let me just think the best way so I'm not rambling too much. I think, okay, so the carbon constrained hypothesis, right? So if we think about AM fungi, and how they colonize a plant root system. These are obligate symbionts, right? And they require, maybe you might say, well, they're all about the carbon. So this is what they need. This is why this is for them. This is what they're after. And so when it comes to things like community assembly, or like which fungi are within the root, it makes sense that, well, the amount of carbon that's available to the fungi is going to impact the fungi, right? That's their resource that they're really looking for, that they tend to be limited in most of all. And so when we think of something, or all the different things that might affect how much or what kind of carbon might be available to the fungi, then things like insect herbivory or pathogen infection, these are big, these are huge factors that can affect plant productivity and plant survival, of course, as well. So it makes sense that this will have a big impact on the amount of carbon that's available to AM fungi. And so the carbon constrained hypothesis kind of really focuses on that, right? So this idea that for a long time there was, I suppose, a theory, a concept, an understanding that things like insect herbivory or pathogen infection would reduce the amount of carbon going to AM fungi. Although there actually wasn't a lot of, or any real sort of empirical evidence to demonstrate that really well until recently, from Katie Field's group at Sheffield. And so the idea here is that, well, in terms of the community assembly of AM fungi in the roots, if you have a herbivore, a pathogen that's reducing the amount of carbon, it's this change in carbon availability that's going to be the key factor determining the AM fungi that are gaining membership of the community in the root. And so if you're then limiting the amount of carbon because the plant has less carbon available or is directing the carbon elsewhere, then it's going to select for particular fungi that are suited to low carbon. On the other hand, the defense-directed hypothesis is this idea that, well, it's not only about the carbon, because we know AM fungi can enhance plant defense. And there's some evidence that suggests this might be an evolved, heritable trait or function. And so is it then possible then that the plant would select particular fungi either for enhancing their defense, or that the defense mechanisms within the plant have a strong role in allowing or selecting particular fungi within the root system? So then it's more sort of giving more recognition to the defense function of the fungi and how that is likely to influence the communities of AM fungi in the root. So I suppose that's my very quick summary of those two theories.
Yeah, cool. No, that's super interesting. Cool. Yeah, I guess are you able to elaborate on the specific mechanisms that the herbivore plant interactions, how they affect the AM fungal assembly?
Well, we don't, I suppose, know for well, those things haven't been tied together quite nicely yet. Well, as nice as they should be, maybe. But so we know, for example, things like jasmonic, so the plant defenses, right? So there's lots of different plants have different defense mechanisms or defense strategies that they rely on to be able to deal with antagonists, to deal with pathogens and herbivores, and they're different depending on what kind of attacker that is. So we tend to see that for when plants are attacked by certain pests or pathogens, that we get different defense pathways are upregulated. So we have what you might call or what we do call jasmonic acid phytohormone. That's associated with particular defense pathways against particular antagonists. And then we have salicylic acid pathway, which is a slightly different defense pathway, again, for different types of pests or disease. And this is more sort of induced defense response. So upon attack, the plant has this biochemical response that's quite complicated, and many people's careers have been focused on unraveling that. But we know that jasmonic acid and salicylic acid have an important role in the AM symbiosis. So this is where this link comes in. And we know that when people have looked at, okay, how do AM fungi increase plant defense when they do, there's a lot of evidence to show that this is to do with the sort of priming of plant defenses and priming of these hormonal pathways, particularly jasmonic acid. So plants that are colonized or have engaged with the AM symbiosis, when they're attacked, they have a stronger, I suppose, reaction, a greater defense reaction to attack than those that are not colonized by AM fungi. So there's this sort of defense priming effect that the AM symbiosis has. Yeah.
Cool.
And so there's this link between those hormonal pathways between the fungi and the pests.
Yeah. Okay. Is there any evidence that there's particular groups of fungi that upregulate the jasmonic, or I'm going to not say this right, the salicylic acid?
Yeah.
Upregulate those pathways? Are there different groups that kind of do different bits
Yes.
distinctly, or?
Yes. So there's evidence that it is different for sure. So even back before we look at, so if we take a step back and even just look at like the outcome of defense. So work that was done a long time ago was looking at, say, comparing one AM fungal species or taxa, one group of taxa versus another. They would say, okay, well, isolate this, and this has a greater defense response, has a greater defense outcome for the plant than this group of fungi, this group of AM fungi. But people weren't looking at the mechanism then, but we just knew that the insect, if it was an insect herbivore, the insect would perform worse, so it would not grow as well, or it would avoid the plant, or other things, or the plant could tolerate that better. So we know there was differences between different AM fungi. And then more recent work has looked at has found the same variation. So certain AM fungi will have a stronger effect on jasmonic acid pathway or the salicylic acid pathway. And this has been done both at the sort of metabolite level and also the transcription level. So looking at the gene transcription. So I will say as well that it's more that we see upregulation of jasmonic acid with the establishment of the symbiosis. So it's more about upregulation of that particular pathway. And there's a lot of evidence to show that this varies between different groups. Whether we can say, oh, this family of AM fungi is very good at this, and this family of AM fungi is very good at that. We do say that, but it's actually it's not based on the comprehensive, literature that you might think it is. It's very much just based on a couple of studies years ago, and now everyone just says that as the dogma. So
yeah,
I think a lot more studies just to clarify that those kind of groupings is really important.
Okay.
Yeah.
So just so I understand the defense-directed hypothesis correctly. So the idea is that the plants selecting for these groups that help upregulate these jasmonic these defense pathways, but also that the fungi also need to be kind of adapted to these lower carbon kind of input levels. So it's kind of like a little bit of both going on in the hypothesis, or?
Well, it could be. So that I don't know. I don't know anything, I suppose, but that is unclear because it could depend. So it could depend on a number of things. So if the plant decides, or if the plant is allocating whatever the mechanism is, there's more allocation of carbon to a specific AM fungus that is very good at enhancing defense. Maybe actually might require more carbon, but it's
Yeah.
worth it for the plant to invest in that in this instance.
So it's
But
more about the potentially that the plant is just selecting where it's putting that carbon. So it's not then the actual fungi that's upregulating these genes isn't necessarily getting less carbon. It might be that the plant's directing more carbon to that fungi.
yeah,
Yeah.
that's one potential scenario. But
Hmm.
it could be even as that's a sort of very strong. So I guess at the most extreme level of the defense directed hypothesis, it would be that the plant is selecting particular AM fungal taxa that are effective at particular defenses to deal with particular antagonist, and that the plant is directing, even if it has less of a capacity to fix carbon, it directs that carbon towards that fungus because it is worth engaging in this symbiosis for the benefit it gets. So that's at the very extreme
Yeah,
level. Uh, wouldn't that be interesting if that was the
that'd
case?
be really cool.
But I suppose at a weaker level, it would be maybe it's just the fact that um, the complex changes in the biochemistry of the plant as a result of being attacked that just influences the assembly of the AM fungi. So it's less of a I'm picking you for this particular outcome. It's more a we're getting different fungi in our roots because, as a result of some complex biochemical changes that are affecting the 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 fungi that are remaining within the root. So that's a sort of weaker interpretation of the defense directed, and that
Mm,
the defense does have an influence, but it's not quite so specific or directed,
yeah.
I suppose.
Okay.
So all those things could be possible, I think. And yeah, it'd be cool to test all those, I think.
Mm. Um, I guess, well, I've got a question. I know you work in predominantly in agricultural settings. So I do wonder, do you think it'll be one day it'll be feasible to implement AM fungi as an effective pathogen or herbivore defense tool at the farm scale?
Do I think it's possible?
Hmm.
Yes. I don't think it's possible now. Um, because it just seems so cliche, right? Every ecologist says we just don't know enough and it's too context specific. But I think it is possible, yes. I maybe don't think it's so feasible that we'll be able to. I mean, well, maybe, but I suppose it's, in my opinion, it's less likely that things like applying inoculants is would be the way to go, at least on its own anyway. I think you
Mm.
would need to, it'd be more about the right sort of habitat for the particular fungi that maybe are particularly good at enhancing defense to encourage their establishment and persistence in your system. But I think before we get to that stage, we there's just so much of the, as I see it, fundamentals that need to be figured out. So,
Mm.
like I mentioned before, we think, Glomeraceae are particularly good at this, whereas, Gigasperaceae are particularly good at that. And it's nowhere near as simple as that, I think. And we just need some better data on how different, at least even just at the higher taxonomic levels, maybe just at the family level, we can see is there are these uh, defense enhancing traits? Are they uh, grouped within specific taxon groups, or are they not? And if they are, how is that? Well, we need to know that first,
And I guess
and
it's
then
also about how that it also probably is a bit of a plant dependent question as well. So different crop types might need different groups of AM fungi or different species of AM fungi.
mm,
Mm,
yeah, I think it would, yeah, because it would also you would also from realistically you would also need to take into account the plant benefit as well, right? So it might be good at defense, but what does it mean for yield if it's a crop we're talking about? Ultimately,
mm,
that's what's going to matter in a lot of ways, in a lot of circumstances. So um, that's maybe a different aspect of that symbiosis.
cool. Well, I think it's probably a good time to start wrapping up. So I thought I would just finish up by asking you, um, You'll just bring up your paper. So you finish up with this box 1, which is a number of kind of outstanding, I guess, future directions for this research. And I was just wondering if there's any specific directions that kind of spark your interest, or any areas that you intend to kind of move towards as your next steps.
I don't want to give too much away, so but I think, well, in terms of the most like critical, or what I see is like a really important thing that maybe might be boring for some people, but is that sort of first question in the box, I think, like, is the variation in the AM induced plant defense, is that actually a result of selection rather than just drift? Is it can we say that it's an outcome of evolutionary selection, and that it's to do with, you know, the benefit that plants have, this other benefit that plants have garnered from AM fungi? But yeah, I think all those questions, of course, are very really interesting. I also think for everyone else that's working a lot on, you know, fungal traits, I think that's really interesting. So maybe it might not be so easy to see a signal taxonomically, but maybe there are particular AM fungal traits that we can associate more clearly or more quickly with defense enhancing outcomes for plants. So yeah, that's a pretty interesting question, I think.
Cool. All right, it's probably a good time for us to finish up. Thanks again for joining me today for the what we're reading, Adam. And yeah, I'm really excited to see where you go next with this.
Thanks, Luke.
All right, I'll see you next time. Right, how do I stop this recording? I'll just have to cut it. Oh, there we go. Stop.